Hello everyone. Today we are going to learn viral infection and its pathogenicity. So what is pathogenicity means? Viral pathogenesis. It means the study and process and the mechanism by which viruses cause diseases in their target host. So it's a process and mechanism how viruses will cause the disease in target cell, how actually the disease is caused. That's what we're going to learn today. So in this chapter there is 4 main parts. One is infection and its disease, and then after that mechanism, after that types of infection, what kind of infection is there, and after that our immune response, what our host body's responses towards the infection. The first one, viral infections. What are the infections are there? Number of infections are there, right? In the head there is encephalitis. All these viruses can cause encephalitis. Then in the eye, infection. Then there is common cold. In the nose, rhinovirus is the most common one. And then after that there is, what is this, pharyngitis in the nose. Then after that there is gingivostomatitis, that is in the teeth. This virus can cause. Other parotitis should be in the cheek. And then pneumonia, cardiovascular hepatitis, skin, myelitis, enterogastritis, lots of places where virus can cause disease a lot. So the thing here, history little bit. So at first in 1997 smallpox, and then after that eradication of that, AIDS started. Then after that mad cow disease. These all are viral infections. And then outbreak of hantavirus, Ebola virus, and then right now there is COVID. Virus. It's not only right now, it's like going on around. And then what are the mode of transmission is like, first it will enter the body, then it will multiply in the body, it will get out from the body, and it will enter in another body. Normally germs is there, and also can we how to define this chain of infections, like germs are there, virus, bacteria, parasites. It will enter into the body. It is present in animal, dog, food, soil, and then after that it will come out through what, the sick persons, mouth, cut, and then how it is around, like contact, droplets, how it will get in, portal of entry is mouth, cuts and eyes, and then next sick person go again. See, routes of entry, how can viruses enter in our body are like from skin, if it is cut it can enter, oral cavity, respiratory tract, and then digestive tract, food, and then genital tract. Mainly sexual contact. Now let's study what is tropism is. Tropism is what? Tropism is the ability of a virus, given virus, to productively infect a particular cell. So what is the character of virus is, it will just affect one particular cell, tissue, or host species. It doesn't affect the whole body, right? For example, this cellular tropism means HIV affect macrophages, neurons. Influenza virus affect lungs, brain. Myxoma virus affect in human. This is host tropism. This is tissue tropism, like in rabbit, human. On what things it depends? It depends on these three things: receptors, what kind of transcription method is there, what cytokine determinants means, what kind of like antibody, antigen is there, kind of thing. Depends on that. Now let's see transmission in the body, like what can transmission. One is chupu. This is localized infection, meaning virus will spread in between the organs, whereas meaning viremia means it will transmit around, like through the blood, using blood. It will flow into the blood and then transmit. In the generalized infection, what is generalized infection? Generalized infections occurs through nerves. Generalized means it is one part of the body to another part of the body, not only organs but parts. And now viral pathogenesis. Now let's learn pathogenesis. There is two parts. One is virus will directly infect the cell, whereas one virus will suppress the immune system and thus causing the injury in the cell. So directly cause, first one is cell death. For example, polio virus will cause the cell death. Cell will die. Cellular proliferation for molluscum contagiosum. What is this? This is cell uncontrollable growth will happen in the cell, causing tumor, and then malignant transformation, again uncontrolled growth, and then causing cancer. whereas in some cases steady state infection means it will slowly grow slowly, and then at last death happen in the interval of many years, like 20-30 years. These are all direct cause of the viruses. Yeah. Further, we can say it as a cytopathic effect. CPE, very important. What is cytopathic effect? Let's see what is cytopathic. Cytopathic effect refers to the structural change in the host cell. Structural change in the host cell caused by the viral infection. So what happened? Some changes occur in structure. For example, change in morphology will happen. What happened? Cell rounding. Different kind of cells are getting rounding. Degenerate. Something will get lost. Aggregation, loss of attachment to the substrate. It will, in the surface it will not attach. It will start floating. Histological changes means in the histology inside what changes occurs. Inclusion bodies, will come into the body. What is inclusion's body? Inclusion's body are aggregated of specific type of protein found in neurons, number of tissue cells including red blood cell, bacteria, viruses. Okay, this is one definition. Whereas another is inclusions are those aggregates of the virus particle. See, so some virus particle will enter into the body. Basically, that is what it means. Now another is synthesia formation. What is synthesia? It is a process of formation of multinucleated giant cells. So there are lots of cells which will come together and then fused, and then there will be lots of nucleus. That is what synthesia is. Then change in cell surface. Then in the surface of the cell there will be different antigen will get expressed. This is the picture for like from 3 to 48 hours when different that cytopathic effect is shown. Now what is cytocidal effect? It is like basically this picture. See, what happens is a virus will enter into the cell and then enter, and then what happened? Uncoat, translation, formation of lots of proliferation or lots of bacteria is formed, and then it will release, breaking the cell. That is all processes cytocidal effect. That's what cytocidal effect is. Now what is steady state infection? Steady state infection is like remember I said like what happens is that virus will slowly grow inside but doesn't cause quick death or cell apoptosis, and then but in long term it will obviously happen. Cell growth will happen, virus growth will happen, and then it will lead to the death. Normally it occurs in enveloped palm or enveloped pinto, whereas this cytocidal effect, this occurs in like naked virus. Yeah. Now this is an example of like how virus will enter into the body. This is a chromosome of DNA, capsid will form, and then one virus capsid, and then spike will form in the cell surface, and then it will through the process of budding or exocytosis is called, I think, and then it will go inside. This is how virus enter into the cell body. Now next is HIV. What is it shown here? It is like there is one gene GP120 in the HIV. It will attach with CD4 cells, and then attach with again other lot of them get attached, and then multinucleated giant cell will happen, which is also called synthesia. The one which we learned in the front beginning. Okay, now about inclusion's body. What is it says about inclusion body? Inclusion body is like our cell surface has what? Some are eosinophilic background, some are basophilic, right? There are different kind of cells. That is what we will see. Along with that, we will see some inclusions body, extra stuff which are not supposed to be there in the cell. Those are called Negri's body or inclusions body. For example, in the during the rabies virus, if rabies virus affect our body, then we can see some Negri's body. We can see like this Ni Chi Xiao Ti Negri body. Where we can see in this hippocampus cell and then Purkinje cells. Yeah. So that means like if rabies virus, you got rabies, like how rabies is got. You got rabies you'll get rabies by like when the animal bite, then you'll get rabies. At that time the doctor will search for this Negri's body is present in hippocampus and Purkinje cells are not in your body. That's what doctor will search. Now cell apoptosis. Cell apoptosis means what? What will happen is when virus enter the cell, then cell has this process by which it thinks like, oh, I am the problem kind of thing, and then it will kill itself to protect other cells. That's how it's stayed, right? Also, that is a protective response of the host. Yeah. Now next one is integration. What is integration? Integration is like viruses will integrate into the host genome, and then it will be there for the long period of time, and then slowly it will cause cancer. Integration method chunk. It normally occurs in circular DNA. See how it occurs: human papilloma virus and cervical cancer. So this is a host chromosome, human chromosome. Let's just say this is a papilloma, that is viral infection. This one, now this one will get integrated with the chromosome, and then it start growing, and then this is how tumor grows. This is how tumor happen, and then leads to cancer. Now after that, we need to know about what is this. That was the direct one. All this one we learned was direct effect of the cell. Now how does how relation of our immunity and then viral infection is like? There are two types of immunity we have: innate immunity and adaptive. In innate immunity, we already have the antibody for some certain viruses, right? So what happened is, like, when virus attacked, our body will activate those antibodies, and it will kill that infected cell. Whereas in adaptive immunity, we'll get antibody from outside, we'll acquire acquired, and then after that, when virus attack, it will again kill that infected cell. That's how because of our immune response, our cell will die. It's not direct, but yeah, cell will die though. This is how it works. This is what is explained. Okay, after that we have to learn types of infection. What are the infection type of the virus infection type? There is two types, basically: inapparent and apparent. Inapparent means also called subclinical, which means this is asymptomatic. No symptoms will be seen. We will not know any symptoms. Will be just fine looking from outside. Whereas in apparent infection, we'll see the symptoms. In acute infection, there are different types of apparent infection. First one is acute. Acute infection is like, okay, there it is very symptomatic, symptoms will appear. Then persistent infections, there is another part called persistent infection, which is again divided into many types. Inside that, the first is chronic infection. What is chronic infection? Chronic infection is like, you will carry the virus, but symptoms will be like, will shown like slowly, like you cannot see any symptoms in the beginning, but there will be cell damage kind of thing. So basically, no one will know you are carrying the virus. And in the latent infection, so after this process, after the chronic infection, latent infection will come. Here you start showing clinical symptoms also, and then inside cell organs will also get damaged. For example, this herpes zoster virus or VJP virus, in this kind of virus. Whereas slow virus infection is like, the incubation period is so long, you will not even know you have that infection, and then the incubation period is so long, and then later it will start showing symptoms, and then progressive until it will die. For example, in HIV people, the HIV, you will not even know, if you have HIV. Sometimes it's like that. This is a graph how it is shown. In the acute infection disease episode means the cause, for example, common cold. You get infected with common cold, for example, rhinovirus, and then symptoms will be seen and you will get cured. That's what it is. Chronic infection, little bit disease will come, and then it will not be symptomatic, like, it will just be inside. Latent infection, some disease will come and then it will diminish, and then no infection, no demonstration will happen. You will wake the carrier, but later, at very late, again symptoms will come. Whereas slow infection is like for HIV or something like that, prions. Slowly it will rise. Infection slowly will come, and at last until you die, kind of. Yeah, basically it's like that. There is an classic example of HCV, hepatitis C infection. What happened is, hepatitis infect our liver, right? Yeah. So liver will be normal. At the first acute infection, some pain will happen, and then the pain will disappear. After that, you will not know. After that, chronic infection will happen. That means slowly infection, no symptoms, infection. And when infection reaches around 80%, then it is chronic hepatitis. At what time you'll start seeing symptoms. Then after that, cirrhosis and cancer start happening, and then after that cancer develop, cirrhosis will happen, and then your risk will increase 1 to 4% per year slowly. This all happens within in 20 years or more, so it takes so long time. If it is fast, then it is in 20 years, but if it is in slow, it is 30 years, so it is long process. So how it is transmitted is like there is two way of transmission. One is horizontal transmission, one is mother to child transmission. Horizontal is one person will transmit to several people through different way, like through air, water, fecal route, blood transfusion, those kind of route. It is most common. Whereas in mother to child transmission, it is like mother will transmit to the child. So this is not that much of what is this called, profound kind of this is more available, or what is it? Yeah, you understood, right? This is less. Now let's talk about immunity of our immune. Immunity is two types of immunity are there: innate immunity and acquired immunity. Innate is the one which is already present in our body, yeah, whereas acquired is like we'll get it from the outside. That is acquired immunity. Innate immunity. And again, immunity can be divided in active and passive. Active is the one which is already present in our body, whereas passive is we get from someone. For example, when the child get the immunity from mother, that is passive immunity. Yeah. Next one. Now, to protect from this, what is there in our body, right? Basically, there is many things, many barriers to protect from viruses and other infections. Some of them as like skin. Skin has a main barrier, right? Skin. So unless and until there is a cut, nothing can enter from our skin. That's a good thing. One. Another is inside. Let's just say it went inside the body. Now there is another barrier called blood-brain barrier. Where what happens is like if our blood circulate all around the body, yes. So blood can carry a lot of things. Fruit, protein, sugar and everything. Whereas if let's just suppose some virus goes into the blood, so what will happen? It will travel into the brain, but there is blood-brain barrier, what will happen is like in the brain, it will, it's like a door. It will selectively open something, some necessary stuff, and other it will change. It will again let go along with the blood. It will not enter into the brain, so virus will not enter. That's how another protective method. Another is placental barrier. What is placental barrier? Is like it is a barrier where like only mother which is a tube to connect like a child and a mother, but in first 3 months it is not fully developed. So that's why sometimes the parasites, bacteria, viruses can enter before 3 months, and then it will bring some change in the body of these children. But after that, it's not possible to enter, I think. Yeah. So it is also one kind of barrier. Okay. So how does our immunity work? Immunity works is like what happens. So immunity, that is like to target this antigen. Antibodies are present. This is a classification of. So basically, immunity is provided by WBC. WBC is divided into three different cells. I think inside that there is one called lymphocytes. Inside lymphocyte there is T cell and B cell. Those T cells are the one who is provide immunity to our body. Provides to our body. So how does that work? And then there is another thing called macrophage, so in WBC, which also activate the immunity. How? Let's learn this. The virus is there. Let's just say virus in our blood. Then macrophages will know, like oh, there is a virus, and then macrophage will engulf the virus. Then what will happen after that? After that, macrophage will activate T cell. And then what does T cell do? Two things. One is T cell will activate. There will be B cell. B cell will form plasma cell. Plasma cell will form antibodies, and then antibodies will attached with the virus, which is also called antigen, and then it will eliminate through our body. One. Whereas another one is helper T cell will activate the cytotoxic T cell. Then cytotoxic T cell will kill that destroy the infected cell. Wherever the virus is there, those cells they will infect, and then one and another one is make antibody for those viruses. So both way can be, killed. Okay. So what type of antibody is present in our body? And antibody present in our body is like called immunoglobulin, which is glycoprotein. Basically, there are different types of glycoprotein. For example, IgM, IgI, IgA, all of them, it comes in different situation. For example, IgM is like when the infection is just started. At that time, IgM appears. So if IgM is seen in our blood, that means okay, there is infection started. We should know like that. And there is IgI. IgI is appear where it appear in like blood and tissue. So if there is infection, remember if there was infection in placenta, when that mother and child before 3 months, IgI can enter inside there and kill those viruses, bacteria. Whereas what does IgA do? IgA is like like, for example, there are some bacterial viruses which can exist in our digestive tract, right? Although it is basic acidic in nature, it can exist there. In that case, what to do? In that case, this IgA, this antibody can go there. Also, it can like protect, like, it can also exist in the breast milk. So that's why like children should be given breast milk for 6 months for immunity. Yeah. Now, what is opsonization? Opsonization is like, let us say what is opsonization. Opsonization is immune response which use opsonins to attack foreign pathogen by eliminating phagocytosis. What is it by this? This means basically there is one protein called opsonin. After this protein is there present into the body, then it helps us understand that okay, this cell is infected, kill this cell. This cell is infected by virus. Now it's time to kill. Yeah, that cell. That immune process is called opsonization. Okay, now there is a graph of how does immunoglobulin work. First of all, antigen appear in our body. For SBV, hepatitis B virus, the example is given as, yeah, and then it will increase. At the time our antibody is also formed. It will also increase. Antibody is also increase, and then antigen will start decreasing. Antibody is steady, right? Antigen, EGM, to protect from HBC antigen. Antibody is formed, IgM. Then it will also decrease, and then this one is for later one, like if it occur again, like again, like it will produce anti-HBS. This is for later. Yeah. Now in summary, what is there in summary? First of all, know the concept significance of virus affinity, and then know what is CEP, cytopathic effect, yeah, and those structural change, and then cytocidal effect. Virus will enter and then come out from the cell. A lot of viruses will come out. Then steady state infection, that it will grow slowly for a long time, and then this multinucleated giant cell, that synthesia, a lot of in HIV, lot of cells connect, and then it will make multinucleated giant cell. Then this inclusion body, Bauhanti, is like in the rabies there is inclusion body called Negri body, from the virus particle, like that. And then apoptosis is cell death. Cell will die thinking like it is harming the body. Integrated infection is like in the host cell, virus will get in the DNA or chromosome of a host cell, virus will infect, and then it slowly cause cancer or something like that. Whereas now in the types there is Mansing, which is like chronic infection. Chronic will grow slowly, little bit of symptoms. After that, latent where symptoms occurs, but and then last one, viral infection. Yeah. After that, transmission mode, one is horizontal mode, one is from mother to children. Whereas antibodies is IgM, IgA, IgA, IgM. In which condition, which kind of immunoglobulin is formed, we should know that. Yeah, that's all.